name is Harrison. I've had a really modest upbringing. I was born and bred in a small village known as Rurie in Nyandarua County, a place around Kalau. It's grown a little. My education started at that place, a small school that was then known as Rangers View. And this gave me an insight of what I really needed to do in the future. After finishing my primary school in that place, where the most enjoyable thing that I enjoyed in that school, what really grounded me was the science cafes. I think then they were being called, they were science-based cafes. I can't remember really, Science Congress here. I later joined high school, Nyandarua High School. Really interesting. It was a mixed school then, national mixed school. After 4 years, I joined university where I joined Kenyatta University. Initially, I had purposed to be a doctor, but after studies and going to those clinical routines, I think it became a bit boring for me. I needed something that was not, what can I say, like routine work. So I delved into research and I did graduate with a biomedical degree. I further pursued bioinformatics in my master's, which I concluded around 2012. Then I joined my PhD program after working with different companies in Nairobi University, which I finished sometimes in 2019. And I've been working remotely for a company, maybe you guys have heard of it, which is called Bioinformatics Consultants, where we do consultations. I think that is the brief background of my academic and simple upbringing up to this point. As a kid, we used to play these games with my siblings. So we used to act like doctors and try to do research. We would go out there, plant new plants. But I want to say, like, I've walked this path through my, maybe my mentoring, where I looked up to my grandfather, who also is known as Harrison. And he used to be this herbalist who used to know this specific plant treats this does that. And we did this when I was little. And I used to work with him in the forest, and we used to carry these barks of trees. So I would really say when I was really little younger, that was in primary school, and this kept on growing. So I would just put it as that. It's that early age when we were growing up in school. When I go to the university, I think I had a very clear conscience after attending the first I think anatomy and physiology classes. We were viewing mocks when we joined med school in KU. Then it had just started. Over time, I realized, okay, as much as we are a physician and we are just treating conditions, I wanted more. That budding interest that I had since I was small to offer solutions, I think pushed me into biomedical research, where I did my biomedical degree then. We went for internship where I went to Coast General, and I realized as much as we would be able to be physician and write these, what do you call them? Request forms, you could take a history of a patient. Sometimes a patient and getting the good results, there's some research that is entailed within that description. So I think that led me to go to that end where I was interested in just offering these solutions. And when I was doing my undergraduate, I got very good mentors. I can't say I'm here just by myself. I was mentored by a very good friend who I don't know if I can mention, but he's called Dr. Gashara, who inspired me to the field of bioinformatics, because then it was something new. And there was this computational element that was being brought into research. And I was very good in computing. So it was a field that he inspired and guided me to further pursue. And Then I think we went online with my friends. It was not something that was being spoken about when we were finishing our undergraduate. So we had to sit and maybe research and check, where can we get this degree or these skills in terms of education that can in future propel us to whatever we want to achieve. So thanks to Dr. Gashara, we were able to do a simple check, and the universities or campuses that are offering Bioinfo was Sanbi in South Africa. Then we had the Center for Biotechnology and Bioinformatics as a center of excellence in Nairobi University, and that's where we went to do my master's. After finishing my project, my thesis, which was specifically on some bioinformatics, where we were able to predict structures of macromolecules, I grew interest in drug design, molecular dynamics, and more computational work in terms of simulations. And that is what I pursued for my PhD, where I still did bioinformatics, specifically on neglected tropical diseases. And I was trying to find, can we find intervenes or ways we can be able to alleviate the problems that we have as a country, and specifically as Africa. We have these diseases we don't talk about too much, not very well researched. And if they are very well researched, sometimes you'll find there are loopholes in terms of drug discoveries. And maybe for some which have drugs that have been proposed or have been used for a long time, you'll find these drugs have a very detrimental effect. So that is what I was trying to achieve when I was working for in my bioinformatics PhD project. And we were able to find some better interventions where we can improve in terms of drugs for this neglected tropical disease. I think that is my journey when it comes to simple biomedical research and where we are at. After graduating, we went out there, and there were these, once you graduate with your biomedical degree, there were really big challenges. Because I remember then we had this board, I think it's called the Kenya Medical Laboratory and Technicians Board. And its purpose was to regulate research, specifically lab research in a medical setup. I think that was a big challenge, where you have learned all this, then you're told you must sit for another exam from that board. So I think most of the people are discouraged in that. And when you look at the employment history of most of the graduates who finish with biomedical, there is a big challenge in terms of getting work. You'll find you are again defined to work in a lab setup in the hospitals, which take you again to that routine call. So I think once I finished my undergraduate, getting a job was fundamental for me. But again, I was looking at, do I really want to squeeze myself into that space that was created by that board? So that becomes a bit challenge. So what we did, what I did myself was ask good questions through mentors. So these mentors were really important. I maintained that network between my lecturers, and they would guide, they will tell you, this is at this point, this is at this point. And I managed to get a contact, and I worked for some times in a lab that was in town that was known as Fields Lab. And my advice to any young man is, please, the networks that you grow within your institute are really important. So that's how I got my first job, which I think was a bit lacking some guidance through my university networks. I wouldn't have gone much, even in my research and my work right now, if I didn't get these people who held my hand. And I think this, when we were growing up, maybe they were not called mentors then, but I'm thankful to my parents. They were able to give us this exposure by introducing us to maybe people who are doing different things in life, teachers, you'd meet somebody who is an engineer, then at some point you'd emulate. And I can't push it too far. My grandfather served a very important portion of my life in terms of mentoring me to do biomedical research, because he was really key to it. In my undergraduate, I had several people who held my hands, and I'm so thankful to them. I mentioned Dr. Kashara, who was really instrumental into what I am right now. Then I went further, which he directed me. He told me, Harrison, I was mentored. There's somebody who held my hand. I didn't know stuff, but they held my hand, and whatever I know, I owe it to them, because they were gentle with me. They taught me basic things. So he helped me during that early academia and research. Once we finished school, he sent me to somebody, and he told me, please, this was my, let me call him academic father, because that is the guy who supervised him, Professor Wallace Bulimo. And I remember him asking me, young man, just come to the lab and see what we do in the lab. So I can say this is fundamentally important, and it keeps you guided and focused into seeing what the country maybe does and what is happening within research institutions within the country. Bulimo now, and you see, these are steps to step. Then Bulimo was able to tell me, okay, Harrison, if you want to do bioinformatics, I am not in Sebib, but would you talk to somebody? And he gave me Professor Mullah. I think I've known him for like over 13 years now, and I wouldn't say that there is any point he let me feel like I wasn't enough. He kept on challenging me, and he's held my hand since then. But also when it comes to other things, I can say you can find an array. He, through his networks and students that he mentored, or he would refer you to, would you talk to this? And I'm glad I was able to meet somebody who really helped me during my master's project, Dr. Fred, who was in the States and was working then by a small company that was known as DNA Software Inc., which was very fundamental for me. And you can see through his network, he connected me to a student he taught a long time ago, who took me in and was able to even show me how to do a project. I think this was important in my academic life and the researcher where I am at. He even taught us to write. You know, sometimes you do these packages and you think you can write, but at some point when you're mentored, how that art of scientific writing, it's totally a different ballgame. So that guy was patient enough in making me learn some things that are really fundamental for any science person or any person who is seeking to write any article into a journal. So I think that is important, and I think these are the small bits that are not taught in school. I'm still getting mentored. I'm new. I'm an early career researcher, so I still get mentored. I also offer mentorship to young students, not only in the university where I teach, but also young people who are in high school and primary school. Whenever I meet them and I get a chance to speak to them, and I cannot overemphasize in research or any career that you choose, having mentors who tell you the real challenges that are existing in that field, giving you the broad outlook in terms of national issues, in terms of aligning yourself in wherever you want to end up. But most importantly, and I think I tell my students this when they're getting into their first years, like as much as you come to school, please sit yourself down and really understand what you're doing here. So it will become more meaningful, even attending classes or even shaping whatever you want to do in the future. So I think that is very key and fundamental. My first project that I got involved in was some study when I was finishing my undergraduate, and we had to do a thesis. The first project I got to is some epidemiology study where we were checking rates of diabetes and hypertension in, then it was not Nairobi County, it was Nairobi as a municipal council in a city. So I got the feel of data collection, then refining that data, then analysis. We use these statistical softwares, and you can see this kept on growing my interest when it comes to data. As much as we get that primary data, what can we draw, or what are the inner things that you would find from this primary data? So that was really my interest, and that was something that I really needed to understand. When I joined my master's, and when I came to choosing my project, I worked with again Plasmodium, and as I told you, DNA Software Inc. had come up with a software that is known as RNA123, but they had tried to understand the structures of macromolecules in terms of, for example, the ribosome, the Golgi operators. And from their understanding was they are just chemical elements, and the way they combine, can we really tell how they combine? So them being one of the first companies that was trying to solve the crystal structure of different organelles, it became really interesting. So I was involved in a project where they had just finished assembling that software in all these small bits to be able to predict the structure in terms of theoretical structure. Then they had not done, they had not used the software to do anything that was eukaryotic. They had done only for prokaryotes, but they had refined it for E. coli. And when I came on board during that project, I was trying to test the software in terms of where can we just put these thresholds, what can it do, how big can it predict. And I did the first project where I was trying to predict the structure of the 40S ribosomal subunit for Plasmodium. Remember, really interesting project where we would sit now with all this data that is theoretical, then just find a probable template. We would look at, we know how the 40S is, can we use that to build our mold in terms of this new this new structure that we want to predict. And I remember when I finished that project where we had predicted the structure of the 40S ribosomal subunit for Plasmodium falciparum, I remember a team that had predicted, had crystallized and published their work. I remember in 2012, the Ramakrishnan group in the UK received the Nobel Prize for predicting this, obtaining the first crystalline structure of that. So it was really interesting for me to be able to predict again the structure of that 40S ribosomal subunit for Plasmodium falciparum as the first, because after we did our publication, after that project, the people who are crystallizing, who are doing the real lab work, obtain their structure. And if you check in terms of resolution, our structure is still supreme. So it tells us, as much as we had these databases that have primary data in terms of all the molecular work that is done, you extract the DNA, then you can know, once you annotate, you can know all the regions. You can pick that information and just thread in, and you can be able to find really quickly how the structure would look. Once we did that, I did complete that project. I was involved in another project where we were producing enzyme or designing enzymes from bioinformatics elements. Then we were able to clone them. This, I think, was my second project. Then we can be able to express whatever was required. And I remember working in the lab for a long time. And this was a fundamental thing where I think during my mentorship, people are not getting very well what bioinformatics is, but sometimes I tell people bioinformatics is a multidimensional approach to research problem. And I tell people I'm not from a computational background, though I'm good at it. I'm from a biomedical background where I can get in the lab and do this with science stuff. So that really exposed both these strengths, where you can design a project in silico using your bioinformatics skills, then go to the lab and implement it really simply, avoiding so many mistakes and so many misuse of some of the reagents. After that, I got into another project, which I was working on for my PhD. And I was looking at, again, Now, since you can predict these structures, how far can we take this in the future? And I remember there was this call by medicine for malaria venture. And then they were trying to find, okay, we have this expression, can we be able to screen? And I'm sure most researchers would understand the process of screening, or you have maybe a metabolite or whatever is your lead compound, then you must do these cultures, then you expose to your cell line, for example, then you test the activity. If the growth is happening, if there is a limitation of growth, that can be a good antibiotic depending on whatever you're doing. So if this can happen, it is very difficult to produce a drug really quickly. So what I worked on was I checked on a multiple thing, and I'm sure they are called kinetoplastids, the likes of trypanosome and leishmania. So I worked on obtaining the structures for all the kinetoplastid, that is trypanosome, gambians, brucei, and cruzi. Then I did for leishmania major. The reason why I did this, I was trying to compare. As much as they are classified as kinetoplastids, and if you look at their DNA, they are closely similar to each other with minor deviations. But when it comes to the diseases they cause very different diseases. So I picked that as my study, where I did a prediction of the ribosomal structures. Then I was able to find specific portions within it, which we call the motifs, which can be used as drug targets. And we did some publications on that, where we obtained and we were able to, again, do a methodology of how you obtain this. And we managed to do several papers, and we filed a patent in terms of methods of predicting 3D structures of what we call it, nucleotides, in terms of either DNA or RNA. I've been involved in other projects that involve synthetic biology, where again we sit, and my portion within our lab setup is I design everything in bioinformatics, and I keep on querying. If we have, for example, to order primers or probes, how do we design them? What is the best way to do it? How, for example, do we attach a specific, for example, a compound that would be an indicator? So right now I work on those. I also help students, which we supervise, or anyone who comes in and really has bioinformatics challenges within our field or within our lab, that I come in. So in terms of projects, I think those are what I'm doing right now. I'm also interested in predictive elements, where as a side, we were working on trying to find some things that would help in terms of treatment of what we're having as COVID. Because as you find out, when we are doing these predictions, during our project, COVID was not there. So COVID has really accelerated how we did things and how we thought like we would be able to offer remedies for some of these challenges that are existing in terms of diseases. So we've worked, we've done the structure prediction of COVID as the whole organism, where we model the whole the whole RNA, and we can add the specific structural proteins around it. Then at every point we can challenge using drug leads. We can just download everything from PubChem, then write small codes, then you can be able to see this this binding, and you can then check the activities and what happens if this would have in terms of it being used as a drug molecule. And we do this simulation. So we are still doing that, and I think our publication will be out soon. I think one of the biggest challenge any budding scientist, for example, who ventures in science, I think I'll say this: once you finish school, the challenge, one of the biggest challenges is getting a job, because there is either regulation for you to get to that point, or some point you'll find in terms of science, and for example, bioscience, there are very few industries that are willing to offer you jobs. I remember the only jobs that most of my colleagues would get after our undergraduates were medical representative, where you go, you learn about a drug, then you go pushing it into either pharmaceuticals or even to doctors to prescribe it, which is like you've become a sales and marketer for that specific drug. So one of the highs is, one of the lows is getting to that point. Then you really understand what you want to do, but getting an avenue where you can maybe learn more, maybe equip your skills where they are really needed, you'll find that that is a bit low. So you'll find most of biomedical researchers who venture and go and do their master's projects, most end up either becoming university lecturers. And you'll find there is less money of establishing a well organized lab. So I'll tell you that is a big low. You'll find most of the graduates, even postgraduate people, finish, then if they cannot be absorbed by the mainstream research facilities that are within the country, you'll find you have challenges with them in terms of them getting a place where they can work and earn a living. So that is a low. Some of the highs, I'll tell this. I'm one person who believes in small wins. I'll tell you, it's very stressful being in the lab, specifically when you're doing a project and you keep on having these failures. So I'll say, I always tell and encourage people small wins. If you wake up in the morning and you've set up an experiment, for example, and you go and get good results, that is progress. And I always make sure I have this, let me call them small highs at every point of my scientific career. I've had very good wins. Actually, when you have supervisors and once you get to a point where you know what you're doing, because your supervisors don't guide as much as they help you during your research. Nothing is brilliant like them telling you you've become an expert of something that we ourselves would learn from you. So I think that is another high that I have, where your mentors and your supervisors are like, wow, you are able to do that at that point. So at every point of my science journey, I've had those small wins where I felt confident that I've helped solve a problem. Then the best way, once you solve this problem, can you be able to communicate it to as many people? And I think this is the intention of these open access journals or articles that you write. So that is wonderful. I always feel whenever you send a manuscript and it's accepted and published, those are again small wins. And for a scientist, very major win. So I think those are some of the better things. Another high that I can mention is being able to work with researchers, students. I think this is something that I feel gives me so much pleasure, where you get to see them ask questions, know how to ask these questions, and learning how to go about answering these scientific questions. So this I think there is much progress. As much as there are these challenges that are existing within the market, you can see progress. I would bet you've seen progress with many people working remotely. As much as sometimes scientists, you must be in the lab or within a research facility, you'll find sometimes working offline and where you learn different things. We've opened up like conferences or knowledge avenues so much within that science space. I would say these are positives that have happened. And I think for new researchers, I'm really happy to see the government has implemented something where it is giving out the research fund. So for any undergraduate or any student or even any person who is out there, there is that innovation thing that is ongoing. I'm sure you've heard about the new agency, Kenya, and the new established National Research Fund that is channeled through NACOSTI. So I think for any student, if you asking a really good question, because these questions are asked by all students at some point, there is a way you can attract some funding that would help you conduct that research and get it out there to be useful. I think that is one major thing that I would ask any person who is delving in research. Please don't be quiet about whatever you're doing. As much as you think it's really small, it would be of major importance to the people that you are doing that for. And I always tell people this, specifically my students, whenever I'm in class. The reason why maybe you come to school is for you to be able to offer or to impact the society where you come from. So you've come here, you've learned science, you've understood some basic parliaments of science. So the next thing is make sure your science is, or the way you're able to articulate things, you can sell it to the basic people. If I always tell them, if you can sell that science to your mother, if she understands what you can do, she can recommend you to somebody else. So as much as you're doing this science, let it be of impact to your scientific community or even your community wherever you are. So that is something I'll tell them. Always pursue these avenues. There is so much, for example, grants, and again, this is another platform that has come up, and I'm really excited with the spaces that just graduates are able to even communicate the ideas really easily. The social media platform has become very important in terms of communicating important information when it comes to science. For example, on the issue of funding, now you'll find many scholarships, many fellowships, many grant application processes that are advertised online, and you'll see for scientists or small groups of scientists, you can see them form small, we call bionets, where you can always channel this information through, and whoever and whatever they're doing, you can come together and collaborate and write something. Something else that is of importance for any person getting into this space, don't underestimate the process of collaboration. And I'll go back to that point where I was saying, when you're in school, you must understand, why am I doing this? And again, in that school setup, always encourage my students, please always know what you are very good at. And you'll find that becomes a drive. You can be able even to do things as a group. And this is always encouraged, where you sit as a group and have a discussion. Maybe somebody is very good in anatomy, another person is very good in physiology, another one is very good in biochemistry. So you can see in the future as you go on, if this guy goes and becomes very good in anatomy and he ends up in anatomy lab, you'll find at some point you need to collaborate. And I cannot overemphasize this process or this main point. Please don't forget your peers. Sometimes we fall short when I say this. You can work in the lab, but you forget the basic people who are in that lab, the people who we call technicians, who's helped you during that trouble. So make sure you come together and collaborate. So I think those are the key fundamental things that would be important for any person who seeks to get into biomedical research. I think this is common emphasis within the lab and within every conversation that you hear. Research sometimes can be really stressful, as you've mentioned, and that stress can lead to even bigger problems in terms of depression or even major things. I'll say this is because it becomes lonely, specifically when you're thinking things in your head. As much as your supervisor is pushing your agenda, you must listen to the different supervisors or the different team members. Then you must be very articulate in what you are supposed to do. So for any person who is pursuing the course, again, I'll tell you, it's challenging, but I'm sure everybody who pursues it has that heart and that head to take care, to see it through. But I'll say this, I've seen half of the students who get in biomedical research. I think the statistics for the country is half. So I think it is important to make sure, even if you have these stresses, I always tell people, and I've mentioned this a lot, please always see the small wins. If you're able to go to the lab, set up an experiment, run it, even if it gives you a negative result, that is a result that you can document and say, at least I got a result because I was hell-bent to do this research. So I always encourage small wins. Make sure you take this array. Again, make sure you're getting the right people. Don't pile so much on yourself. Sometimes I understand most students have different challenges. You have where you're coming, there's that family element. you're in the lab, your supervisors are pushing you, they need you to maybe get your publication out. You're not getting the right person to talk to or to understand where what you're doing. So I always encourage, and I've said this in my prior statements, like I would encourage a lot getting together, having an activity that you do besides, like I'll say this, as much as we love to stay in the lab or our research facilities, it's good to have something else that takes you out of it. But don't forget to celebrate your small. If you can achieve this, go celebrate. Always ensure that you can talk to anyone. And this is something that I encourage students to do, or any person who is in the lab. Make sure the person who is supervising you or your mentor always understands your science and understands what space you are at. Always let the family help out at some point. Some of the critics that would give you the best critique in your research are the people who you think would not be interested. So I always encourage you have this diverse conversation with different people who would pick it, and this is family and close friends. So maintain that along. Then I think this is important. For example, in the lab that we make it deliberate. If you see somebody is having troubles, if you cannot help at that moment, I always encourage take breaks that are very structured. Like, I'm having these challenges. Don't stop stressing yourself. Like, take a break, go rethink about that thing, sit, do something else, rejuvenate, and then get back to your main course. That is something that is really key. If you have bigger challenges that you think that are stressing you, which are not lab related, I think don't feel any shame to find a counselor or somebody that you can talk to in terms of confidence. Get them to understand your situation and they can help you. Don't die alone. Don't cramp your head so much. So I think that is important. From where I stand, I think the future is I can say the future is just the future. All we have to is make sure you set a pedestal where you can mentor or you can guide as many people. And I always believe on this saying that says you're never successful until your successor succeeds. So I'm at the point where I feel I can create and establish a platform where enough people in my field would find me as a stepping stone. The future is bright, as I've set up or I've said before. For any person who is coming, including us, we can have this conversation, meaningful conversation in small groups, which some time back was an exclusive club for very few people. So I'll say science is very important in our country. We're expanding that reach in science. It is still not enough to see as a country, science and innovation and research is in a good plane. But I would say this. For me, I'm still curious to learn. I learn every day, and I'm really interested in, as much as I learn, I love to teach. So I think I'm in a good standing when it comes to the future and what it holds. And I can foresee bigger things coming out in terms of real tangible research outputs and outcomes that can help not only the country, but the continent and the globe at large. I think it is important to note that I think as much as we castigate social media for our social engineering, I think social media for anyone who is doing research can be a very good platform to communicate, to be able to share experiences, to be able to share some common knowledge that would be redundant whenever you're doing your work. I think this would be a very good platform, and I'm always encouraged to see anyone who gets into social media specifically with an intention either to push good information or good research, and to even ask questions. And I think this is a good standing where we are at and how we're exploiting all this technology that has come to us. I think bioinformatics caters a lot. For example, if you have a specific strain, I'll give an example, for example, for COVID and whatever we are mentioning in terms of variations. I think at any point, you'll find any disease because of the DNA and how it's interacting with the environment. You'll find there are some places it will mutate or change. Importantly, when this mutation happen, the virus is trying to either evade something or hide its epitope, or the portion that is important in terms of causing it disease or propagating it for it to be able to survive. So what Bioinfo can do, you can see at every level, you can do an alignment and tell at what rate is it changing and at what position is this happening. So that will be one indicative how bioinformatics can solve that problem. The next thing where we can use bioinformatics, we can implement many data analysis and algorithms that would tell you, for example, at what point, and this is a really good question that you've asked, at what point can we use a drug or abuse it until we have a mutation that would be detrimental? I think in that forum, you can see how Bioinfo is helping you not only see how this resistance is happening, but you can be able to even predict in the future, if this is used or abused, what are we able to see? I'll mention a study that I was involved in. For example, tuberculosis. Most of the drugs, vancomycin, was working on the RNA, and at some point, that position within that ribosome component that the drug used to bind to and caused disruption of growth of that specific cell, that portion mutated. And now, using bioinformatics, we can really tell what changed within that. A single nucleotide changed, and now it became resistant. But we went further into looking at that specific vancomycin, and there's a team in South Africa that has been able to activate that vancomycin to again be a very effective drug against tuberculosis. So in all that, I think Bioinfo serves at every portion where you can have not only this primary data that you have derived in the lab, you can be able to find more information within that data set by using bioinformatics and data mining skills and tools that you've studied in bioinformatics to be able to offer that solution